Welcome to this exciting tutorial about the process of meiosis.
Today we will be showing you the details of this process. Yeah!
The process of meiosis starts with interphase. This phase is before meiosis actually begins, but is necessary to the entire process because during this time, everything important begins to copy itself. In prophase 1, the two centrosome areas that include the centrioles begin to migrate to opposite sides, or poles, of the cell. Spindle fibers form between the two centrosomes, and the nuclear envelope begins to break down. Throughout this phase, chromatin condenses into chromosomes. Also, the very important step of crossing over occurs. First, each chromosome matches up with its homologue, and they stay next to each other. A chromatid from one chromosome is aligned with a chromatid from the homologous chromosome so that the genes for a specific trait on the first chromosome are aligned with that of the second chromosome. These spots on the genes are then broken by proteins, and a synaptonomal complex begins to form, which attaches the two homologues. Then synapsis occurs, which is when the breaks in the genes are closed because they are joined to the corresponding part of the genes of the non-sister chromatid. When synapsis is over, the synaptonomal complex breaks down, homologues drift away from each other a little bit, but they never fully leave each other. Now metaphase 1 begins. All the pairs of homologues line up along the symbolic metaphase plate in the middle of the cell. These pairs are all then attached to spindle fibers, specifically at the kinetochore. As anaphase 1 begins, the homologous chromosomes are separated from each other by proteins and pulled apart by the spindle fibers. The pairs of sister chromatids stick together and are pulled towards the same pole. The sister chromatids or the homologue move towards the opposite pole. Telophase 1 starts out with a haploid set of copied chromosomes. A cleavage furrow begins to form in animal cells. During cytokinesis, the cytoplasm divides and makes two separate cells. Depending on the species, the chromosomes will decondense and a nuclear envelope will develop.
Meiosis 2 begins with prophase 2. Two daughter cells are present in prophase 2. The chromosomes are condensed and centrosomes are duplicated. The centrosomes gradually move towards the poles and the chromosomes begin moving towards the metaphase 2 plate.
Metaphase 2, the chromosomes are lined up at the metaphase plate. The kinetochores bind to spindle fibers from opposite poles.
The third step of meiosis 2 is anaphase 2. The sister chromatids gradually move toward opposite poles. As the chromatids are moving toward opposite poles by the spindle apparatus, the poles move further apart as the dividing of the cell begins.
The last step is telophase 2 and cytokinesis. The chromosomes reach opposite poles and cytokinesis takes place. The final result is 4 haploid daughter cells with 23 chromosomes each. Since crossing over occurred during meiosis 1, the 4 daughter cells and parent cells are genetically different from each other.